Our lab for a long time has been working on this concept of dormancy because it is very clear evidence, not in the experimental level, but at the clinics, that the disease is capable of being apparently cured for months, even years or decades. Unfortunately, patients relapse later on. Hello, I'm Héctor G. Palmer. I'm the head of the Stem Cells and Cancer Laboratory at Vall d'Hebron Institute of Oncology in Barcelona, Spain. So mostly we collaborate in the PERSIST-SEQ consortium by working on colorectal cancer patients and specifically trying to answer one of the biggest questions, that is why patients acquire resistance to some treatments. Study a small population of cells, considered persister cells, that tolerate the treatments and later are responsible for the progression and relapse to the treatment. We are one of the few academical partners that are bringing some samples coming from patients recruited in clinical trials here at Vall d'Hebron Hospital. We also work on preclinical models like organoids and patient-derived xenografts. In all cases, cells derived from patients. And more particularly, we work on acquired resistance or persistence to treatments against oncogenic mutation that is known as BRAF. That is a population of 10-15% of all colorectal cancer globally. Being part of the PERSIST may have two major advantages that are very, very important for us. One is, of course, to collaborate with other scientists and industry in a very high standard on the scientific level and very focused on translating the results to patients' benefit. Second is technological advantage. Single cell evaluation at the level of RNA-seq or other type of omics is helping to understand better this complex phenomenon like persistence and acquired resistance to different treatments, because we get more detail on heterogeneity of the tumors. Not all the cells within the tumors respond the same. Within this heterogeneity relies part of the mechanisms of resistance to the treatments that we are trying to improve. So we go from patient samples, patients treated in clinical trials with BRAF inhibitors, comparing pre and post treatment, using organoids with cells derived from these same patients, also treated with the same drugs, and see how the cells persist. We also use the same cells but injected in animals, in mice, and we see how the cells persist and later progress and acquire resistance. And finally, we also develop a transgenic mouse model that is BRAF-mutant, that we can evaluate there not only the intrinsic persistence or drug tolerance of these tumor cells, but also the stroma, because these are immunocompetent models. By today, we've been able to sequence samples from the first three technical scenarios: patients, organoids, and PDXs. Soon we'll have the ability to do equivalent experiments with the transgenic mouse models, in which we are very excited to also include spatial transcriptomics in this scenario. Thanks to integrating all this data, we are really understanding how the cells persist to these specific drugs in colorectal cancer. We are learning a lot on the single cell sequencing and starting to understand mechanisms that nobody had previously described before in this specific scenario of BRAF-mutant colorectal cancer. PERSIST-SEQ mostly is using the technology of a single cell, studying omics of different kind at the level of the single cell. We haven't yet used other omic technologies that are available, that is on the epigenetic level of the cells, or proteomics. Depending on the first round of results, we may propose to use other omics to integrate with the first round of results to deeper understand the mechanisms behind the persistence. Dormancy is a very peculiar state for a tumor cell. Anybody knows that a tumor cell is designed to divide in an uncontrolled manner, acquiring many alterations. How is it possible that it's just entering these phases of complete latency? So we have been working on how the cells manage to enter in this state. It's important because these cells, as they do not divide, they are intrinsically resistant to most of the treatments that are designed to kill dividing cells. But also we discovered that this resistance to drugs is not only passive because of no division or not proliferation, but active. So they express many of the genes and proteins required for eliminating the drugs. So all this is important because the final aim is once you get in a scenario where you may not be suffering of a disease, but you could be suspicious to have high risk of relapse, we could actually target these undetectable cells by inhibiting or blocking some of the processes that make them viable in a dormant state. So with the final objective of really cleaning the body from all the latency cells, so you will get maybe final cure of the patient. Most of our models in the laboratory are generated with cells derived from patients, either patient-derived organoids or patient-derived xenografts. One of the best examples to understand how our models are suitable for answering the main question of PERSIST-SEQ would be those that are generating a metastatic disease by injecting the patient-derived cells in the case of colorectal cancer in the intestine of mice. They generate a primary tumor. Later on, they generate metastases in the liver and lungs or lymph nodes that are those organs where in patients metastases occur. So it is quite beautiful to study now, thanks to the PERSIST-SEQ, how the primary tumor may use not all the cells present in this first lesion, but just subpopulation of cells that are prepared or more fit to generate a new metastasis, and how that happened under the pressure of a treatment. We have been exposed to the discoveries of very excellent laboratories in which we are together understanding the biology of these persistent cells. We have been exploiting our collaboration with other researchers in the field of colorectal cancer and in the field of these new technologies for proteomics with single cell discoveries. And all this is making our projects to accelerate not only the discoveries, but the understanding of persistence. Our lab is translational by nature and also is our institution that is based in the largest hospital in Spain. And clinical trials in oncology department is one of the most important at the world level. Most of the big companies are developing first-in-class drugs here. The results of the PERSIST-SEQ in our particular site here at the Department of Oncology will be easily translated to the clinic and could be ended in an academic clinical trial. Give you an example. We work on BRAF inhibitors, and for a while we've discovered some of the mechanisms that confer patient resistance. Thanks to the PERSIST-SEQ consortium, we can go even deeper on the understanding. And now we are realizing that not all the cells, but a population of cells is responsible of this acquisition of resistance. These cells do have peculiarities that could also become new targets for the future. That new drugs or repurposing drugs used for other diseases could be rationally combined with inhibitors, for example, for prolonged benefit of these combinations in the future. I'm personally very excited to be part of this consortia, being able to interact and collaborate with other researchers in the academy, but also in the industry, and exchange ideas and not only learning about persistence, but also about science in general. It's very exciting discussing with some of the brilliant researchers in the industry because it's always focused on translating the results into the clinical practice. But also it is a very great opportunity to exploit all the beauties of single-cell omics. It is something that we have been doing in our institution, but not in collaboration with some of the most reputed experts in the field. So we are learning with the best in the world in these technologies, something that we have not yet here in our institution. And it's a great opportunity to share the discoveries with brilliant people working either in the academy or in the industry. So, and focus for later translation of the results into a patient's benefit. We are so focused now to complete our proposal that is quite ambitious. We want to sequence cells coming from different models, but all of them trying to answer parts of the main question of persistence against BRAF inhibitors in colorectal cancer. We envision that before the end of the project, we'll be able to have a very solid understanding of this through the single cell sequencing. Once the consortium ends, makes all the data available for the scientific community, we may be one of the most important databases in terms of single cell RNA-seq and other omics that will be the base for many other lines of research, not only from the current members of the consortium, but for any researcher that may have a hypothesis. They can then later navigate through the data that the consortium has generated and made public for everybody.